So just remember that double fertilization is the concept which is found only in the angiospermic plants. That means it is found only in the flowering plants. And a double fertilization concept is said double because there are two male gametes and both are going to participate in the process of fusion. Now one of the male gamete fuses with the egg and the other male gamete fuses with the two polar nuclei of the central cell. So one of the fusion will give rise to the zygote, the other will give rise to the, zygote is diploid in nature, and the other male gamete will give rise to a triploid structure called as the primary endospermic cell. Because if you remember, there are two polar nuclei, each are haploid. So already there is n plus n, and when a male gamete enters, it becomes n plus n plus n. So there are triploid situation now, and this cell is called as the primary endospermic cell, which will later develop into the endosperm, and zygote will develop into the embryo. Such questions are oftenly asked in the exam. Now please be sure that this diagram is the diagram of a fertilized embryo sac, because I have seen students drawing very awkward or manipulated diagrams or their own designed diagrams. So you need to follow and read NCERT carefully. NCERT clearly says that this is the diagram of a fertilized embryo sac. So you can see that there is a surviving big nucleus, and this central cell has now become primary endospermic cell, and this polar nuclei now have become triploid nucleus, and it is the primary endospermic nucleus. And then you can see that it is clearly mentioned here that the antipodal cells are degenerating, and on this end it clearly says synergids are degenerating, and only surviving cell on this end is the zygote, which is diploid. So this should be your diagram if it comes in the exam to draw fertilized embryo sac showing the result of double fertilization. So double fertilization has given rise to PEC, and PEC carries a nucleus called as PEN, and then the other structure which survives in the double fertilization is the zygote, which is diploid in nature. So this should be your answer. Now what is the significance of double fertilization? If it is asked, then you can mention that double fertilization ensures that the nutritive tissue, or which is called as the endosperm, is formed only when the formation of the embryo has been confirmed by the fertilization of the egg. If generative fertilization does not occur, what is generative fertilization? The fertilization which gives rise to zygote. Then if the zygote formation does not happen, then endosperm should not form, because there is no need of energy to be provided to any structure. So double fertilization's second significance, it provides characteristics of male plant to the next generation, as well as provides nutritive tissue also. So that's because the male gamete is going to participate in both fertilization, the syngamy as well as the triple fusion. So in both the concepts, male gamete is going to provide characters, and then fusion of the second male gamete with the polar nuclei provides stimulus to its cells to resume growth and form the nutritive tissue called as the endosperm. So endosperm formation only takes place when the second male gamete fuses with the two polar nuclei. Remember, pollen tube, when enters into the embryo sac, into the ovule, it may enter maximum time. NCERT says it enters from the micropyle, but yes, there are some exceptional plants in which it may enter from the chalazal end, or it may even enter through the integuments. When it enters from the micropyle, it is called as porogamy. When it enters through the chalazal end, it is called as chalazogamy. When it enters through the from the integuments, it is called as the mesogamy. So now the next thing to understand here is this diagram. You can clearly see the zygote shown here. This zygote divides into two cells, and it is you can see clearly it is a asymmetrical cell division. So one cell is small and the other cell is big. So this bigger cell will continue to form a suspensor, and the smaller cell will continue to form the embryo, or you can say the initials of embryo, followed by the embryo and then a mature embryo. So these diagrams are important from the point of board exam, because what they do generally is they will ask you to draw from the zygote onwards. So first you have to draw a single cell situation, followed by two cell situation, and then a globular proembryo, followed by globular embryo, followed by the heart-shaped embryo, and then you will have the mature embryo. If you go through the NCERT, you can check these diagrams are clearly mentioned. And please remember, sometimes they will ask you to draw the situation which is before mature. So we remember the sequence. After globular, you will have heart shape. After heart shape, you will have the mature one. And please remember, suspensor is a structure which suspends the developing embryo, and it even absorbs the nutrition from the endosperm and passes it on to the embryo for development. So these are the things mentioned in NCERT. You can check here, syngamy is mentioned, triple fusion is mentioned. So all these things are clearly mentioned in the NCERT. Now the next thing to start is the post-fertilization structure. So after fertilization, what happened? Don't say zygote formation is post-fertilization. Zygote formation is the result of fertilization. What happened to this result is called as the post-fertilization event.